Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, we are in a continuation of the chapter that is transporting plant. Okay, so now we are at the last lecture approximately. Okay, and we are going to discuss today the how the phloem transport occur, how the phloem play a role in food transport from the source to the sink. So phloem, we know that mainly that is involved in the food transport. And here the flow occur from the source to sink. So source is the region where synthesis occur, and the sink is usually the region where the storage occur. But it is not that source sink cannot be the source. Sometimes source and sink can be reversible. So what are they? We have to discuss in detail. Let's discuss it. So it is a flow from source to sink. So food, primarily sucrose, is transported by the vascular tissue phloem from source to sink. So first we remember the synthesis of a food occur in the form of glucose. Then from one plant organ to the another plant organ, the transport through the phloem occur in the form of sucrose. And when the food getting stored, it is stored in the form of starch. So the flow from source to sink will occur. So growing tissues are usually act as a sink, means they need nutrients. And source is the region where the synthesis is going to take place. And here you can see in the presence of light and CO2 the food synthesis occurring. So leaves are acting as a source. So usually the source is understood to be the that part of the plant which synthesizes the food. And usually these sources are to be the leaf, and the sink is the part that needs or stores the food. And that particularly the root act as a sink, and other young parts which need this particular component or food. But it is not that the source and sink cannot be reversible. The source and the sink may be reversed depending on the season, or the depending on the plant need, or the depending on the conditions. So suppose when the active particular plant is actively photosynthesizing, and in that case the leaf will act as a source and the root will act as a sink. But if the food is not able to plant is not able to synthesize this food, so at that time the leaves will have a deficiency of the food, whereas the starch is stored in the sugar will act as a source. So it's this time when there is a plant has a not ability to synthesize food, the leaf will be act as a sink and the root will act as a source. So sugar stored in a root may be mobilized to become a source of food in the early spring, when the bud of trees act as a sink. They need energy for growth and development of the photosynthetic apparatus. Okay, so for this the root is going to act as a source. So these are the flower bud and the leaf buds which need the, or they are acting as a sink. So since the source sink relationship is variable, source sink relationship is variable, the direction of movement in the flame can be upward or downward, or it can that can be bidirectional. So source sink relationship is variable, and the direction of movement in the phloem can be upward or downward. So that is a bidirectional. So this way the bidirectional movement can take place from root to the particular leaf, or leaf to the root through the phloem. This is in contrast with that of the xylem, where the movement is always unidirectional. We know that xylem carry out the unidirectional transport, that is upward from root to the aerial parts of the plant. Okay. Hence, unlike one-way flow of water in transpiration, food in phloem can be transported in any direction so long as there is a source of sugar and the sink which is able to use, store or remove the sugar. And this phloem carry out a bidirectional transport. Here in this diagram you can see this is the phloem. It is actually xylem vessel, not phloem vessels. This is a phloem. You should not use a phloem vessel word. It is a phloem where the bidirectional transport is taking place. This is the phloem tissue. This is the companion cells. This is the source cell, and this is the sink cell where there is a required. So see what is happening here. This is the phloem. This diagram actually helps us to understand the mass flow hypothesis which we are going to discuss. Okay. So phloem ceph, if we talk about the phloem ceph, what is there in the phloem? what is the is during the transport, then mainly water and sucrose are there in this particular phloem set. But other sugar, hormones, and amino acid are also transported or translocated through this phloem. And in this loading unloading mechanism will work. And in loading mechanism, we will discuss that how this particular pressure flow hypothesis help. So an experimental setup suggested by Munch that help in understanding the bidirectional phloem transport. So Munch given this particular phenomena, experimental connect, experimental one, and in this you can see this a diagram. This is acting as a source and this is acting as a sink, which is a storage part and leaf. Suppose in this there is a dilute solution and it is a concentrated solution because source is there. Here food is synthesized. Okay, so here food is there. So it is a concentrated. So water will getting absorbed. So water will through this pure water will move from here to here. Okay, and ultimately when water will come, the movement of this both will occur in this direction. Okay, so from water will move from dilute solution to this one, but food which is concentrated here will food from move from its higher concentration to the lower concentration. So for the transport of food or a phloem transport, the pressure flow hypothesis or mass flow hypothesis was explained, and it is considered as the accepted mechanism that is used for the translocation of sugar from source to sink is called the pressure flow hypothesis. So the most accepted mechanism for this translocation of sugar from source to sink is explained in this pressure flow or mass flow hypothesis. And you can see this is the NCRT diagram which is explaining us this pressure flow hypothesis. And if you see this NCRT diagram and if you see the labeling, here if we talk about this, here sugar is entering the sieve tube and water is following by the osmosis and higher trigger pressure is developing. So this is acting as a source. Okay, but and the food is traveling through the phloem. Now here sugar leaves the sieve tube and water follows by osmosis. So here the sugar is leaving from the phloem and it is reaching to the tip of the stem, means here it is a sink, but here it is entering the sieve tube, so it is a source. And here also you can see sugar leave the sieve tube for the metabolism and storage and water follows by osmosis, so here is also it is going to be a sink. So the food is entering here and leaving at this sink, and when food entering the sieve tube, the water is entering the sieve tube, but when food leaving the sieve tube, then food water is also leaving the particular, we can say, the sieve tube. So this here water is entering along with the food, then the solution is forming. So sugar solution flows the region from region of low turgor pressure, means it is moving from the high turgor pressure to low turgor pressure as explained in this NCRT. To elaborate it, we have to discuss further. So how this pressure flow hypothesis occur, that if we want to discuss, we can discuss like this. Suppose this is the phloem and this is in the phloem, this is the sieve tube. So this is a sieve tube, and specifically along with this you will see here presence of companion cell. This companion cell have their nucleus, and here along with this you will see the presence of xylem. Now here suppose this is acting as a source, and source is usually act as a leaf, and suppose this is the sink, which is a root, which is obtained as a storage organ. Now, what happens at source? The food synthesis occur, and that occur in the form of glucose. When glucose is getting synthesized by the photosynthesis, it get converted into sucrose. And in the form of sucrose, it travel to the companion cell, and from companion cell, it is going to enter the which one? We can say the sieve tube element, and this is the sucrose. Okay. Now, when sucrose concentration increases, the solute concentration increases, and solute increases. That source solute increases, so osmotic pressure increases. So water will move from xylem to this particularly a sieve tube element at the source. When water enters, so this is a sugar, okay, and now water is entering. So water molecules are also going to increase. So water and sugar, that is the sucrose, combinedly form a sugar solution. Now here water is more, so at a source turgor pressure is high, and water is more entered, so means a water potential is also high. But at sink, if you see here, at this particular point here, the sugar solution is less, so turgor pressure is less, water is less. So now what happens? Sugar solution will move this side, and here water and the sugar molecules are there at sink in the sieve tube. Now this food molecules will pass from here to the root cells, and they get stored in the form of starch. So now this a movement of sucrose into the sieve tube through the companion cell is known as loading. So at source, loading of sucrose occur in sieve tube element through active transport. Whereas when here sucrose is unloaded at the sink, so at sink unloading of sucrose from sieve tube element by active transport. So when through active transport the sucrose leaves the sieve tube and stored in the sink, now water is left. Now the remaining water will move back to the what a particularly xylem element. So here the turgor pressure was low, and water is sugar solution moved from high turgor pressure to the low turgor pressure, and that is due to the pressure differences or according to the pressure gradient. Because of this we call it as a pressure flow hypothesis or mass flow hypothesis. So what you will need to remember, the synthesis occur in the form of glucose, the loading occur of what? Sucrose in the CO2 element. Loading is through the active transport. Then sugar solution, water follows. Sugar solution moves from higher turgor pressure to low turgor pressure. Then unloading occur at the sink. That is unloading of sucrose by the active transport, and the water will leave back, and this water will be again be used. Okay. So this way the pressure flow hypothesis work. Okay. Now to understand more in detail. So, as we have understood, as glucose is prepared at the source by photosynthesis, that is converted to sucrose, that is disaccharide. Okay. Then the sugar is then moved in the form of sucrose into the companion cells, and then into the living phloem sieve tube cells or sieve tube elements by active transport. So the loading is by the active transport in the sieve tube. So this process is known as loading at the source, which produces a hypertonic condition in the phloem. Obviously, in the phloem, the sucrose is getting accumulated. So at that point, the hypertonic condition is established. So water in the adjacent xylem moves into the phloem by the osmosis, and at source, the particularly water will move in this way, in particularly from as the osmotic pressure build up due to the accumulation of water and the solute, the phloem step moves to areas of lower pressure. At the sink, osmotic pressure must be reduced. Okay, and again active transport is necessary to move the sucrose out of the phloem cell into the cell which uses the sugar, converting it into the energy, that is starch or cellulose. So storage occur at the sink in the form of starch or cellulose. As the sugar are removed, the osmotic pressure decreases and the water moves out of the phloem. Okay, so to summarize, the movement of sugar in the phloem begin at the source where sucrose is sugar is loaded, actively transported into the sieve tube. Loading of the phloem sets up a water potential gradient that facilitate the mass movement in the phloem, because of which we call it the mass flow hypothesis. Phloem tissue is composed of sieve tube elements or cells which form a long column with holes in their end walls called as sieve plates, and through these sieve plates the movement takes place. Cytoplasmic strands pass through the holes in the sieve plates, form a continuous filament. As hydrostatic pressure means turbulent pressure in the phloem, sieve tube increases, pressure flow begins, and the sap moves through the phloem to the lower pressure areas. Meanwhile, at the sink, incoming sugar are actively transported out of the phloem and removed as a complex carbohydrate, that is starch. And this particular thing we have already discussed. So the loss of solute produces a higher water potential in the phloem, and water passes out, returning eventually to the xylem, which is explained in this pressure flow hypothesis. Okay, so we have already understood this. This is a source where the sugar source cell physiological process loading sucrose into the sieve tube at the source, then water follows by osmosis, then the pressure flow, phloem and xylem are coupled in the osmotic system that transport sucrose and circulate water, and the physiological process unloading of sucrose to the sieve tube element from the sieve tube element to the sink, and water goes back to the xylem. So a simple experiment called a girdling is conducted to identify the tissue through which the food is transported. Initially, it was not known that through which tissue this food is transported. So to prove that the phloem carry out a food transport, one important experiment was carried out that is known as girdling. Now in girdling, what is done? In girdling, first thing is taken that one particular plant is taken, and in this the ring of epidermis, cortex, and phloem is removed. Means a ring of bark is removed. Ring of bark is removed in a circular pattern. So when a ring of bark is removed in a circular pattern, that will create a thing that is known as girdling. So to understand that, let's discuss this broader. See, this is the step. Now in this, the particular bark is removed. In this region, bark is removed. Okay. So the bark is removed, and in when bark is removed, you can see this particular region is getting swollen, and this region is getting died by day by day. So bark consists of all the tissues which are present inner to the, we can say, outer to the vascular cambium. So bark does not contain a xylem; it contains phloem, cortex, and epidermis. So we have to write a definition that bark consists of all tissue layers present outside the vascular cambium. Now, on the trunk of the tree, a ring of bark up to the depth of the phloem layer is carefully removed. You can see here that the particularly bark is getting removed by the cutting it in the form of ring. Whole ring is cut. Okay, and this way the whole ring cut is provided to the bark. Now, when you will remove this particular whole ring, you can see here, this is the white is the xylem, or we can, the particular phloem, cortex, and epidermis has been removed. So this is the girdle. Now when we remove this in the stem, in the absence of the downward movement of flow, so here what happens, we have removed a bar, means we have removed a phloem. So the phloem is carry out a food transport, so food will not go beyond this, it will not go beyond this, it get accumulated here. Okay, so in the absence of the downward movement of food, the portion of the bark above the ring on the stem becomes swollen after a few weeks. You can see this portion is getting swollen above this particular girdle. Why? Because food is accumulating here and it is not transferring to this particular region, but water and minerals are going upward. Okay. So what will happen? The upper regions are healthy, but lower regions is not getting, the root region is not getting a food, so it goes on dying day by day. So this simple experiment showed that if the particular girdle food is not, because of this particular death of the root system is occurring, so the simple experiment shows that phloem is the tissue responsible for the translocation of food, and that transport takes place in one direction towards the root through the phloem. That was proved by this girdling experiment, and this girdling experiment was done by Malfigi. Malfigi was the scientist who done this experiment. So Malfigi done this experiment. Okay, so that is all about the, what we can say, the phloem transport and girdling experiment. So what is the, what in the adjacent xylem moves into the phloem by water, actually from adjacent xylem moves through the phloem in the pressure of high flow, that is through the osmosis, and going back also through the osmosis, that is the sink osmotic pressure must be reduced. So the phloem tissue is composed of sieve tubes. Okay, so that is all about the what pressure flow hypothesis and girdling experiment, and that is all about the chapter transport in plant. I hope you understood well. Thank you everyone.